Gas chromatography, or GC as it is called for short, is a technique where you will be able to separate out mixtures and also be able to determine the different compositions of compounds in a mixture. So this is the purpose of our experiment in today's lab. We are going to find out or determine the composition of compounds of both our sample. In the mixture that will be handed to you, the unknown sample. Now, how much amount do we need to inject in a gas column? If you remember your column chromatography, and we had bagged sand, we had bagged alumina, and then we had also had our impure mixture on top. How much sample did we had added at that point? We are going to inject a sample in liquid form. And so we will be injecting anywhere between 5 to 7 microliters. So this is the amount that you need. Now, to give you a vague idea of what is a microliter, imagine a small drop of water on a bench. Imagine that drop. It's spherical in nature. It's just lying on the bench. How many microliters of water do you imagine there are? In that single drop of water, there are approximately 100 microliters of water present. And so you can see we are going to use very, very small amounts to do the gas chromatography. The principle of separation is based upon boiling point differences. What was the principle of separation in your column chromatography? Definitely the polarity difference and affinity difference. For column, we also figured that gravity plays a role, and for TLC, we know that the capillary action plays a role. But for your gas chromatography, it is the differences in boiling points that will help you separate out your materials. The stationary phase lining the column is known as carbovax. It's a very, very high boiling liquid. You do not need to know the structure or boiling points for that compound, but you definitely need to understand the difference of stationary phase in GC and the stationary phase in your column chromatography. For your mobile phase, the mobile phase for your column chromatography were actual solvents which would move throughout the column and get the materials out of the column as well. But in this particular technique, your mobile phase is an inert gas. For example, nitrogen or helium. The choice of nitrogen is also based on economical reasons. One tank of helium costs around $100 or maybe $1,000 sometimes. But one tank, a full tank of nitrogen gas with tax costs around $10. And so nitrogen gas flushes through the column and works as the mobile phase. As soon as you inject your sample, this column is at a high temperature. As soon as you inject your sample, because of the temperature, both the volatile materials will evaporate and become vaporized. Who will convert into vapors first? The one with the lower boiling point will be converting into vapors first and will be getting out of the column first and appear on the spectrum first. The length of this column is usually anywhere between 4 to 6 feet. Again, think about your TLC and your column chromatography. Usually how big your columns can be. But this length is very, very narrow in opening and is coiled in the GC. Once your material exits or elutes out, the detector detects how much amount of each sample is there, and you see a particular area under the peak. If you count the spectra that is shown here, how many peaks do you see? A, B, and C. So there are 3 peaks.